Good morning, good afternoon, good evening, and welcome again to the second in a series of webinars organized by Novosanis. Today, we will be discussing the accuracy and validation of pre-analytical and analytical procedures for HPV and STIs testing of self-collected urine samples. And we will do so joined by two key experts in this field, being Professor Alex Vorsters from the University of Antwerp and Professor Clementina Cocuzza from the University of Milan. Before we dive into this exciting topic, I will go over some simple housekeeping rules for this webinar, and I will introduce both speakers and also Novosanis in a little bit more detail. We will then move into the main presentations of today, after which we will go into any questions you might have during a Q&A. But as said, let's go over some basic housekeeping. To keep this a smooth webinar, your microphones will be muted throughout the entire session. If you have a question, please let us know via the Q&A button at the bottom of your screen. Please don't wait until the presentations come to an end. Simply submit your question and we will dive into those during the Q&A at the end of this webinar. If we would run out of time, we will come back to you on any of the questions that we could not cover after the session. And please also note that the full session will be recorded and that the recording will be made available to you after the session. But first things first, and that is to introduce today's speakers. Our first speaker is Professor Alex Vorsters from the University of Antwerp. After graduating as a bioengineer from the Free University of Brussels in 1989, he followed a postgraduate course on tropical veterinary medicine at the Institute of Tropical Medicine in Antwerp. Since 2001, he has been working as a senior project coordinator and researcher at the University of Antwerp in the Center for the Evaluation of Vaccination. He is involved in several vaccine-preventable disease projects, including the Viral Hepatitis Prevention Board. Since 2015, he is also coordinating the activities of the Human Papilloma Virus Prevention and Control Board. In 2016, he obtained his PhD in medical sciences on the detection of HPV DNA in urine and monitoring the impact of HPV vaccination. Last year, he was then nominated an associate professor and is coordinating a research team that further investigates the use of first-flow urine as a liquid biomarker source for the follow-up of vaccination programs, cervical screening programs, and genital tract infections in women. Our second speaker of today is Professor Clementina Cocuzza. She graduated in medicine and surgery at the London Hospital Medical College in 1987, where she obtained a scientific bachelor's degree in biochemistry. In 1992, she achieved a master's degree in clinical microbiology at the microbiology department of the London Hospital, following which she became a researcher in clinical microbiology at the University of Milan, where she accomplished her PhD in microbiological sciences. Since 2001, she is an associate professor of clinical microbiology at the Medical School of the University of Milano Bicocca. Since 2006, she is also the head of the Clinical Microbiology and Virology Laboratory, which is partnering with the BBMRI Eric Atma Expert Center, aiming to accelerate clinical research with a focus on biomarker verification and validation. Her laboratory in Milan is also an associated member of MIRI, a coordinated platform that is managing microbial resources to support research in biotechnology. Professor Cocuzza's research investigates the natural history and epidemiology of infections caused by oncogenic HPV, the development of novel biomarkers, the evaluation of new antibacterial agents, and the epidemiology of infections associated with antibiotic-resistant pathogens, and the molecular characterization of antibiotic resistance mechanisms overall. And then to introduce myself, my name is David Vasters. I graduated in 2002 as an industrial engineer in biotechnology and in 2004 as a bioengineer in food technology from the Catholic University of Leuven. I started my professional career in the pharmaceutical industry in several positions in and around the manufacturing areas of Schering-Plough and Janssen Pharmaceuticals. In 2014, I then moved to the medical device industry when I joined Biocartis, a molecular diagnostics company. There, I started out supporting new product introductions, after which I moved on to leading their customer service and support department. And last year, I joined Novosanis as their senior product manager. And in that role, I'm now supporting the further commercialization and expansion of the Colli-Pee product family. Novosanis is a Belgian-based medical device company, founded in 2013 as a spin-off from the University of Antwerp. In 2019, it was acquired by OraSure Technologies. Novosanis remains headquartered in Weinigem, near Antwerp, but the Novosanis team is spread out globally and is present in Europe, Asia, and North America. Since its foundation, that same Novosanis team has been working very hard to develop and commercialize Colli-Pee, which is truly the heart of the company. Colli-Pee is a sampling device that allows for a volumetric and standardized collection of first void urine in a volume range of 4 to 45 milliliters. Within this volume range, Colli-Pee is an ideally suited collection device for different application purposes. It can be used for detecting urinary cancer biomarkers, as well as for detecting infectious diseases, including various sexually transmitted infections like HPV, gonorrhea, chlamydia, and others. And with that, I would like to turn it over to our main speakers of today, Professor Vorsters and Professor Cocuzza. The floor is all yours.
So thank you very much, David, for that nice introduction. And I'm really pleased to be present today to present you on the importance of first void initial stream urine and the preservative for HPV detection. So these are my disclosures relevant to this presentation. This is the overview of my talk. I will give a very brief introduction on HPV and then discuss important lessons I learned during the past 10 years when using a urine sample, and with the focus in this presentation on detection of HPV DNA. I'll also give you an example of the Colli-Pee used in a vaccine trial and discuss some additional applications of first void urine sampling in vaccine trials. So I think before starting with the science, I think it's important to know the field in which we are operating, which is the field of cervical cancer, the 4th most common cancer in women. And as you can see, in 2018, more than 300,000 women still died of this preventable disease. And most of the cases, as you can see on this graph, occur in low and middle income countries. The World Health Organization has called to eliminate this disease and cervical cancer as a public health issue. And more than 194 countries already agreed to proceed with that plan. And the target is by 2030 to have 90% of the girls fully vaccinated with HPV vaccine by the age of 15 years, to have 70% of women worldwide screened using a high performance test by the age of 35, and again at the age of 45. In addition, 90% of women identified with cervical disease should receive treatment or cancer care. Now, the use of urine as a liquid biopsy for HPV testing is quite obvious. You can use it to follow up HPV vaccination trials or programs, but more and more data is becoming available that it also will be useful as a self-sample in cervical cancer screening programs. Of course, also test of cure setting is an important application. And now we see with the current pandemic that home sampling becomes more and more important as a solution to overcome the current situation. So the potential advantages of urine: it's non-invasive, it can be done, as I said, at home. It's suited for serial sampling, so there's no impact at all by the sampling on the region from where the sample originates, which is different if you compare it to cervical sampling, for instance. It's potentially cost-effective, and it is a highly preferred self-sample, and it can also be used in younger girls that are not yet sexually active. Now, I started my work with the review more than 10 years ago, as I said, on the detection of human papillomavirus in DNA. And what I found there was that there was a very high diversity in the applied methods and outcomes of the studies. So there was definitely room for improvement and standardization, especially at the level of sampling methods, at the level of storage conditions, and also at the level of sample preparation and DNA extraction. And of course, also DNA detection is an important step in this process. So the first publication after my review was the optimization of HPV DNA detection in urine, specifically focusing on collection, storage, and extraction. And the first in-house trials I did were some spiking experiments where I used HPV plasmid DNA and already got clear results that nuclease inhibitors were very beneficial to recover the HPV DNA. I also used HeLa cells to look at artificial spiked DNA and to detect then the HPV DNA, but it's kind of all artificial settings because we really want to know how our methods are performing in real world, and these are samples, urine samples of women with an HPV infection. And therefore, we were very lucky that at that time we had a therapeutic HPV trial ongoing in our institute. So we knew that these women had a cervical HPV 16 or 18 infection. They had normal cytology, but in addition, within this trial, to avoid giving an experimental vaccine to pregnant women, also the urine sample was requested to do the pregnancy testing. So we had access to real-life urine. With that urine, we set up the following trial. So we asked the initial stream of first void of the participants, which was quite rapidly then, within 30 minutes, transferred to a tube with preservative and a tube without preservative. And then different aliquots were further created to do the processing, as I will explain on the next slide. All samples were kept for 3 days at room temperature. So these are the extraction methods we used. I will give you the example of the arm without preservative. So we used the QIAGEN mini kit to do the DNA extraction. Then we used the Amicon ultrafiltration followed by a QIAGEN DNA extraction. We used centrifugation at 20,000 G for 1 hour, where we then looked at the supernatant as well as the pellet. Those DNAs were again extracted by a QIAGEN kit. And then we used the EasyMac nuclesense automate to process 1 ml of the urine. And then in the arm with preservative, we had the identical different extraction methods. And these are the results. So if we look to HPV DNA in the arm without preservative, you can see that the pellet with QIAGEN extraction gives the highest number of positive samples, and the lowest amount is found in the supernatant. Here you see then what the effect of the preservative was. You see in all arms more samples becoming positive, and especially with the supernatant, we see that 16 out of 24 became positive. But the best score in this setting was the Amicon filtration using a higher volume of urine followed by a QIAGEN kit. And here we see the results for human DNA. Again, best results without preservative is pellet plus QIAGEN kit, and only 5 out of 24 samples in the supernatant were positive for human DNA. And the effect of the Preservative is there also quite important, with all samples becoming positive. So, and these results were confirmed by other groups. And there's the example of results reported by the group of Jennifer Smith at the IPV of 2017, where you also clearly see the effect of a preservative and assuring detection of HPV DNA over 7 days, whereas samples without preservative show an important decrease in the detection. Besides the sample processing and the preservative, I think another very important step or knowledge is the rationale. Why are we using this first void urine? And the key behind it, that it's not about the urine. The urine is a cleaning or rinsing solution. So here you see on the figure in yellow all the epithelial tissue of the genital tract, which includes the uterus. So this epithelial tissue has even a mechanical cleaning characteristics for this cavity. You have the different layers that peel off, and all the debris and degree of exfoliated cells accumulates between the small labia, and when a woman starts urinating, this is washed away with the urine flow. Important to know that the first void or initial stream is different from the first urine of the day or the morning urine. I'll come back to that later. I think these pictures clearly show the difference between the initial or first void stream versus the mid or the subsequent stream. So you see much more debris in the initial stream than in the subsequent stream. So these women received 2 recipients and split their urine void over those 2 recipients. Very interesting are these samples from 2 women that had their periods, and you see clearly that the majority of the menstrual blood, which originates from uterus, can be detected in the initial stream, and whereas in the midstream almost no traces are found anymore. In 10 women where we also asked to split their urine stream, we also looked at the number of copies of HPV DNA versus, so the first void versus the midstream, and you see that in a factor of 4.8 up to 160 times more of HPV DNA is found in that initial stream. And we see the same for human DNA, so much more human DNA between there was 1 woman where this was not the case, but all the others goes up to 20 times more human DNA in the first void, confirming our hypothesis. This rationale of first void was also confirmed in a meta-analysis where it was shown that studies using the first void urine had higher sensitivity for detection of HPV DNA. There was one issue that we wanted to point out in this study, that there was one study that also included the initial stream, but prior to urinating, they asked their women to clean the genital area with alcohol swabs. So clearly removing all the debris that we want to use in our testing. So therefore we answered that with a letter to the BMG where we further explained the rationale of using first void urine. How to put this into practice? So knowing that preservative is important, that the initial stream is important, we at the university have a product development group, and we asked a student for his master project to develop a self-sampling urine kit that allows to capture then the first void urine, to mix it with the preservative, and it should be appropriate to send it also by mail. So these are the first prototypes then of what we now use as the Colipy. I think an important question is also, but you can ask women to just provide the initial stream in a classical cup, and you don't need the Colipy device. So to answer that question, we set up a study. So what's the difference between the colipy and a urine cup? And at the same time, we wanted to answer the question if there was or was not a difference between the first void from the first urine of the day versus a urine sample collected later on the day. Just with in mind having the fact that if in the morning a woman has not gone to the toilet for a longer period, there might be more material accumulated during the night. So we had 33 women who participated. They provided 2 urine samples over 4 consecutive days and alternating the collection method. They sent the samples by post to our lab, and before processing, we stored them at -80. We did a quantitative DNA genotyping assay and used combined generalized linear mixed models. These are the results. So colipy collected samples showed higher HPV concentrations than the cup collected samples. However, when there were very high amounts of human DNA, then the benefit of colipy disappeared. However, in 82% of the samples, this high amount of human DNA was not obtained, showing a clear benefit of the colipy in most of the cases. We did not find any impact on the moment of sampling, so first void urine of the day performed equally to urine collected later on the day. These results have been confirmed by other groups, and it has a main advantage that you can choose the time of urine collection when running a trial or collecting samples. This is just an example of urine testing, and this was a large trial we performed in Rwanda, Bhutan. As a matter of fact, we already did a repeat survey, so each time targeting more than 1,000 girls and having response rates of more than 88%. So a quite well accepted sample. So in conclusion, when using urine sample to monitor HPV infections, I think it's important that it's not about the urine, it's collecting the first void urine, which is crucial. DNA is well preserved in mummies, but definitely not always in urine, so it's keen to have preservative added to your urine to maintain DNA. If we look to the timing of the collection, there is no preference. I want to thank you for your attention.
Okay, thank you very much, Alex, for your nice presentation. So now I would like to present some of our experience on evaluating the accuracy of pre-analytical and analytical procedures for HPV and STI testing on self-collected samples. So just starting with some background, sexually transmitted infections are generally a big public health issue worldwide, with WHO estimating more than 1 million STIs being acquired every day. And some pathogens are particularly important. HPV is certainly responsible for 290 million women having infection every year, but also other pathogens such as chlamydia, gonorrhea, syphilis, and trachomatis are also very prevalent worldwide. And moreover, sexually transmitted infections have been showing an increasing trend in prevalence in the last years. And so here in this slide, we see some of the data from CDC showing an increasing trend for all pathogens. And also what is very important is that combined cases, so co-infections, are also increasing. So co-infections with multiple sexually transmitted pathogens are extremely common, and the other problem is that the majority of STIs have no symptoms or only mild symptoms, so they often fail to present to medical practitioners. And this can have important consequences, both in sexual and reproductive health. Particularly, there has been documented mother-to-child transmissions of STIs, often leading to serious consequences such as stillbirth or neonatal death or other congenital problems. Moreover, sexually transmitted infections represent a major cause of pelvic inflammatory disease and may often lead to infertility in women who have chronic infections. They also increase the overall susceptibility to acquiring other sexually transmitted infections such as HIV, and certainly not least is the fact that HPV infections can result in the development of cervical cancer. So this has brought about the World Health Organization in defining a strategy with a goal to end sexually transmitted infections and all the serious consequences that they may bring about, with the 2030 target, as Alex nicely described, of for HPV, but also for other sexually transmitted pathogens, of reducing the incidence of these infections. So how can sexually transmitted infections be prevented? Certainly education is very important, and vaccination where available, such as for HPV, certainly plays a major role in preventing sexually transmitted infections. But screening programs have been the mainstay of prevention for many of these infections, also in the case of HPV. Prevention by screening allows to not only to identify and treat patients with sexually transmitted infections, so preventing the complication, but also to be able to do contact tracing and treat sexual partners of these subjects with sexually transmitted infections, so preventing transmission and reinfection. And of course, as we said before, screening can also allow prevention of cervical cancer in the case of HPV. And if we focus on HPV, as again, as Alex nicely described, there is been an increased burden. It is a major cause of cervical cancer, particularly in young women in reproductive age. So the WHO call to action to prevent cervical cancer has indicated that the challenge is to ensure that every woman over the age of 30 is screened and treated for precancerous lesion. But it is important, in order to reach this objective, to develop innovative technologies and strategies. This is because even in countries where screening programs are available, such as in most industrialized countries, still cervical cancer cases do occur, and they do occur in women who are under-screened or have never participated to screening programs. So some of the new strategies which have been evaluated worldwide are those that use self-sampling to improve participation of hard-to-reach women to cancer screening program. And in this respect, again, there have been important meta-analysis on evaluating the accuracy of self-sampling as compared to clinician-collected samples, and two important meta-analyses, one published in 2014 in Lancet Oncology, and a subsequent updated meta-analysis in 2018 by Mark Arbin, have shown that the accuracy of HPV testing on self-collected samples can be very good, and if this is associated with PCR-based methods. So certainly the accuracy can be assured by the appropriate evaluation of accuracy in using a combination of collection devices together with PCR-based HPV assays, which have demonstrated better accuracy. And in order to evaluate the combination of collection devices and PCR-based assay, Mark Arbin has published a protocol named Valides, which is a protocol aimed to validate human papillomavirus assays in combination, as I said, with collection devices. Not only for HPV, but self-collected samples have proved to be an important strategy for improving screening also for sexually transmitted, and this is just a few publications that show how also self-samples can be used for detecting other sexually transmitted pathogens. So certainly self-sampling has been shown to be an important way, an important strategy to improve screening, but again, validation is necessary. And so I like to talk about the experience of an ongoing study that we are undertaking in our gynecology teaching hospital, aimed at validating STI and HPV assays in combination with collection devices. And this is a colposcopy study, as I mentioned, so there is a cohort of women referred to colposcopy, which have been studied in order to evaluate the adequacy of self-collected samples in terms of cellularity, to evaluate HPV and STI PCR-based tests on self-samples, to determine the prevalence of HPV, STIs, and co-infections between HPV and STIs in our population, in a studied population. We also wanted to evaluate some of the pre-analytical protocols, and I will show you some results from evaluating 2 different nucleic extraction procedures, as well as two different forms of the Colli-P device, one collecting the first void 20 ml and the second collecting the first void 10 ml of urine. And finally, we wanted to evaluate the patient's acceptability of self-sample collection. So the study protocol, as I mentioned, was set in a colposcopy clinic, and we aim to enroll asymptomatic women referred to colposcopy for a recent diagnosis of cervical dysplasia. So those women who agreed to participate by signing an informed consent, they were asked to collect, first of all, a first void urine sample using the Colli-P device. Subsequently, they were asked to collect a vaginal self-sample using a dry flock swab, which was then transported to the laboratory and resuspended in the laboratory in 5 ml of thin prep. Finally, prior to colposcopy, a cervical sample was collected by the clinician and resuspended in 20 ml of thin prep. The patient following colposcopy was asked to fill a patient's questionnaire on acceptability of self-collection. And going on to the methods, as I mentioned, we evaluated 2 different nucleic extraction procedures by semi-automated platform or automated platform. In the case of NucliSense ECMAG, and where we started from 1 ml of samples of each of the 3 different sample types described. We also compare this to the Seegene Nimbus automated liquid handling workstation. And in this case, we started with 200 microliters of sample. We also evaluated sample cellularity as a means of evaluating sample adequacy by means of an in-house human gene quantitative real-time PCR assay. And we determined the presence of HPV and STIs using 2 commercial assays by Seegene, Aniplex HPV 28 and STI7. PCR plate preparation was also accomplished through automated microlab Nimbus, which prepared the extracted samples for PCR detection, and we used Biorad CFX 96 for real-time PCR. For what concerns the assays, they're both PCR-based assays. The HPV 28 Aniplex assay was detecting the 14 high-risk HPV types, which are the ones of most concern for the development of cervical cancer, but was also in a genotype-specific way, also evaluating 5 probable high-risk HPV types and 9 low-risk HPV types. The second assay was STI7, again a real-time PCR-based assay detecting Chlamydia, trachomatis, Neisseria gonorrhea, Trichomonas vaginalis, 2 strains of Mycoplasma and 2 of Uroplasma as common STIs. So moving on to the results, this is the study population in this ongoing study. So up to now, we've enrolled 245 women. And here we see the cytology grade at the time of colposcopy. So we see a big percentage, a large percentage of women had low-grade squamous intraepithelial lesions, 30% had high-grade squamous intraepithelial lesion, and some women were shown to have ascus at the time of colposcopy. On this side, we see the results of colposcopy. So out of 245 women, 66 had positive colposcopy, 47 required treatment by conization. And in those 60 women where we had a histological or biopsy result, we see that we had 2 cases of cervical cancer and 33 cases of CIN3 and 9 of CIN2. So moving on to the sample adequacy assessment, here we have the 3 sample types, and here we have the mean value of cellularity evaluated through our in-house assay in the 20 ml of cervical sample, in the 5 ml of vaginal sample, and 20 ml of urine sample. And we see that very similar cellularity in the total sample was found, showing that all samples had adequate cellularity, so were adequate samples for further STI and HPV testing. In this slide, we see the same 3 sample types, what we found in terms of HPV detection, divided in the 3 groups: high risk, probable high risk, and low risk HPVs. And we see a very good concordance between all samples. So both urine, vaginal samples showed very similar positivity rates as compared to cervical samples. And in particular, for the high-risk HPV types, which are the ones that are particularly important in cervical cancer screening, we see really very similar, very good concordance between the positivity rates of the 3 sample types. In this slide, we see the detection for each individual HPV type, which was detected by this genotype-specific HPV assay. And we see again in orange the cervical sample, the vaginal sample in yellow, and the urine sample in green. And again, we see the prevalence of the different HPV types, but very similar positivity rates between the 3 sample types, with HPV 16 and HPV 31 being the most common HPV types in our population, followed by HPV 53 for the probable high risk and HPV 42 for the low risk types. When we look at concordance and Cohen-Kappa values for the test agreement between, in this case, urine versus cervical sample for HPV, we see that looking over all the high-risk HPV types, there was a very good concordance, and also this was observed for the individual HPV types. Again, here you see in blue, for each HPV type detected the concordant samples, in green the ones that were additionally identified by urine testing, and in orange the ones that were additionally identified by cervical sample testing. So we see that the majority of samples showed a very good concordance. Similarly, we find for vaginal samples as compared to cervical samples. So again, very good overall concordance with good Cohen-Kappa values for high-risk HPV types in general, but also for the single type, we see a very good concordance rate between vaginal tests, sample testing, and cervical samples. When we, moving on to STIs with the STI7 assay, again, here we see the 3 sample types, and we see the positivity for 1 or more STIs in the population of the 245 women. And we see that in this population, in this cohort of asymptomatic women, there was quite a high percentage of patients who also showed the presence of 1 or more STI, ranging from about 45% to 55%. when we look at all the positive STI patients, we see that 75% on average were positive for a single pathogen, for a single of the additional 7 STIs, whilst 25%, which is not a small group of women, had multiple infections, showing 2 or more of the 7 STIs being investigated. So again, demonstrating that co-infections are common in a specific cohort of women who have cervical dysplasia. When we look at the single distribution of the single pathogens, we see that Uroplasma parvum showed the highest number of positive results in this study, followed by Mycoplasma hominis and Uroplasma urolyticum. Also, Chlamydia trachomatis, there were 5 patients out of the 245 women who were asymptomatic but had concomitant Chlamydia trachomatis infections. Again, another interesting aspect we wanted to evaluate was there any association between the infections by high-risk HPV types and sexually transmitted pathogen. And in this slide, we see the prevalences, the percentage positivity rates of high-risk HPV infections together with at least 1 STI. And we see that again, these are very much more frequent than probably would be expected. We see 37% of co-infections between high risk and STI in cervical samples, and 45 and 42% respectively for vaginal and urine sample. And when we further investigated out of all these positive women, how many had 1 or more of the STI pathogen under investigation, together with high-risk HPV types, we see that the majority had only 1 additional pathogen, so in terms of co-infections, but there were also some women, around 18% to 22% of women who had high-risk HPV types together with 2 other STIs, and even some with 3 STIs, so a couple with 4 STIs in combination with high-risk HPV infections. Further interesting was to evaluate in this group of high-risk HPV and STI co-infections, how many of these women had high-grade cytology, low-grade cytology, or negative cytology. And we found that approximately, well, over 80, almost 90% of women who had co-infections with high-risk HPV and STIs had either high-grade cytology shown in the pink column or low-grade cytology. So they had some kind of cervical dysplasia, and a minor group were shown to have negative cytology. So it seems that the association of these co-infections may be associated in some way with cervical dysplasia, but of course larger studies will be required. Uroplasma parvum, as I showed you, was the most frequently detected pathogen in this group of women with co-infections, and Chlamydia trachomatis, the 5 cases of Chlamydia trachomatis identified in this population were always detected in co-infection with HPV 16, 18, 31, 52, and 56. So having seen the overall results in terms of accuracy and concordance between the different sample types for both HPV and STI, I just quickly wanted to show you some of the evaluation which we performed in terms of the pre-analytical protocols. And here you see in purple the protocol using nucleic acid extraction starting from 200 microliters implemented on the Seegene platform, Nimbus platform. And in red, you see the HPV detection rates using the 1 ml starting volume on the bioMérieux platform. Again, you see very similar positivity rates with at least one concordant high-risk HPV genotype being detected in the majority of samples, over 95% of samples frequently, and this showing that the two pre-analytical protocols were both useful and provided good results. The other pre-analytical comparison that we made was taking two different groups of 50 women. In one group of women, we collected urine using the Colli-P 10 ml device, and in parallel a cervical sample. Similarly, with the second group of women, we collected urine using Colli-P 20 ml device and compared again with the results of HPV testing with the cervical sample. Again, an overall concordance of over 80% was found using both devices, so demonstrating that the similar performance. And when we look at the cellularity of by our in-house assay, in 1 ml of sample collected with each of the two devices, again, we see very similar values, indicating that both collection devices demonstrate a good performance in combination with the C-gene assay used. And finally, going to the results of the patient's acceptability by compilation of the questionnaire, we found that 97.6% of patients would prefer the use of Colli-P Novosanis, the collection kit, for the screening, for participating to screening, and also very good positivity in terms of preference for the vaginal self-collection of the clinician-collected cervical sample. So in conclusion, we can say that preliminary data from this ongoing study on self-collected samples using the devices that I described and the assays, PCR-based assay described, show a good analytical performance of self-collected sample for both HPV and STI testing, and certainly patients have documented a better acceptability to screening using self-collected samples. So validated protocols are important. It is important to validate, but once again, the results of these studies show that self-collection can be a valid alternative to STI and HPV screening tests, and that using nucleic acid amplification test provides accurate testing for multiple pathogens on the same sample that can increase participation to STI and cancer screening and certainly reduce public health costs. And on this note, I'd like to thank you all for your attention. Thank you.
Thank you, Alex and Clementina, for these nice presentations. So let's dive into the Q&A. And maybe let's start with a question coming back on the conclusion of Clementina's presentation. So you mentioned, Clementina, that validation is an important aspect in introducing self-sampling in a routine clinical setting. So in your opinion, and also in your opinion, Alex, can the data that has been and is still being collected considered enough as a validation? Or if not, what would be the next steps in validating protocols for self-sampling?
Okay, I don't know if I can start, Alex. I certainly think this data is important in demonstrating analytical accuracy and concordant results between self-collected samples and cervical samples. Larger data is, of course, necessary for, as indicated by the Valides protocol, where the sample size of estimated number of women to be included in a study which involves also clinical validation is 500 women. But certainly the information that we have achieved up to now show a good analytical accuracy. But as I say, probably bigger numbers are necessary in order to confirm clinical validation. Maybe Alex with the Belgian Valides can maybe provide some further information on this.
Thank you, Clementina. And indeed, I think the number of 500 is not the most important number, but it's the number of women presenting with clinical symptoms, because we want to know that the clinical sensitivity is high enough. So therefore, in the Valides, we compare it with a clinical approved and validated assay on the cervical samples, and then we compare it with the results we see in the urine samples, making sure that we have a relative sensitivity that is equal for the SIN2+ women with SIN2+ lesions. So, and that we have already performed with a number of assays, and we presented already intermediate results at Urine last year, and at this meeting this year at Urine, we will further, together with Mark Arben and my collaborators, present the results on the Abbott assay of this validation, but other assays are being run on these samples too.
Okay, thank you. Clementina, in one of your results slides, you mentioned a comparison or at least cervical dysplasia and co-infections with HPV and STI. And there seems to be a little bit less concordance in between the different sample types. If we're looking at the negative cytology results, do you have any idea on the reason for that that drop in concordance? Because the other data all showed very nice concordance in between the different specimens.
Okay, thank you, David. Certainly, this data is interesting to show some correlation between co-infections between HPV and STIs and their association with cervical dysplasia, and this is an interesting aspect which will need to be further investigated with a higher number of women. Certainly in that respect, urine samples and cervical samples showed the highest concordance. Some of the differences may be due to low viral loads that may be responsible for some of these discordant results, or also the possibility of HPV infections in other sites. So again, this is an interesting finding and needs further investigation, but certainly bigger numbers will allow to better evaluate this aspect.
Okay, thank you. Alex, maybe one question for you. Methylation testing is also quite important, maybe more in the HPV triage testing. Could one also consider the use of urine and maybe also ColiP for methylation testing?
Yeah, definitely, and I hope I try to make it clear at least that it's not about the urine. So the urine is just a washing solution, and it is a genital sample that we are collecting in this way. So what we find in our first void urine is really the material that you would obtain by a vaginal self-sample. And I think recently there was a nice webinar that is broadcast by Urine, where Professor Jack Cusick clearly showed and indicated that urine was equal to a vaginal self-sample for his methylation markers. And he will further discuss this also at Urine. So definitely, I think triage assays on the first void urine sample are possible. We're also looking into that further.
Okay, thank you, Alex. Maybe one more question that came in for you, Professor Cocuzza. You showed a comparison in between the ColiP 10 ml and 20 ml volumes. And what would be your preference, and maybe why would you say that one is better than the other?
I think it's difficult to answer this question. I mean, certainly we showed with the cellularity that cellularity is very good, and the volume required for testing is certainly sufficient in both with urine collected with both devices. So in terms of volumes, certainly the 10 ml collection device would be sufficient, but it may be also interesting to note preferences of women in the practical use of the device in this respect. But certainly they both showed very good performance, the urine collected with both devices.
Yeah, thank you. And maybe also to that point, a second question from somebody attending. How do you deal with liquid samples sent in the mail from screening programs? So I think there the smaller volumes would fit right in, because they are really compatible with postal service. But how would you deal generally with liquid samples being sent in the mail?
Well, I think for any biological samples, mailing requires to have several plastic containers to allow the kind of postage or transport of the sample. So if you use a plastic container, that should take care of any eventual spillage. Moreover, the Colli-P device has got a very nice preservative, as Alex was showing in his presentation, and this allows urine to be stable over the time of transport. So in that respect, a very good stability of nucleic acids also in some studies, which I haven't shown you data on this in the presentation, but certainly we see that and as Alex also showed, the nucleic acids are well preserved with the Colli-P device. So in theory, this is not a major hurdle. Okay. Transport of urine samples.
Alex, any idea how long we could keep urine samples stable in the presence of that UCM preservative?
So what we have seen is that it's not indefinitely stable at room temperature. So I think what's important for the preservative, that it's completely innocent. So you can, as a matter of fact, drink it. It's not very nice for taste, but it's completely not dangerous. I think that's a major advantage of the preservative. On the other hand, you cannot keep it for 4 weeks at 37 degrees, because then you will see degradation. But I think at least 7 days at room temperature is not a problem, but don't exceed. And if you keep it at 4 degrees, you could keep it a little bit longer. We keep our samples when they arrive frozen until we process them, and there we see almost no degradation. So okay. I think it's definitely sufficient to do the transport, and we had several trials where we sent the urine sample package to the women home, and they provide the samples back by mail. So and it definitely works fine.
Okay, and maybe given the view of time, we have time for one more question. Obviously, in these presentations, we've been focusing on women patients. But could HPV also be detected in male urine samples? And is that relevant?
Thank you. So the anatomy of man is completely different than women, especially on the genital tract. So we don't have the same principle that it is the ideal sample for males. So it could be that there's HPV infection on other sites that is not washed away or collected by the urine. So we do see a much lower prevalence of HPV DNA in urine. And if you want to sample men to know all the infections, you need multiple sites of the genitals in male. On the other hand, we do find HPV in urine. So if you say, I only look to urine as a monitoring of, for instance, impact of HPV programs, you can use it, but it's not the ideal sample to identify every infection that is present in the male genital organs.
Okay. Thank you very much, Alex, and thank you very much, Clementina. I'm afraid that we've run out of time for today. There are still some more questions that we weren't able to answer today, but for sure we will be coming back on those after the webinar. And please also note again that this webinar was recorded. We will share that recording also with you within a few days. And leaves me nothing else but to thank you all for your attention. Thank you.
Thank you.